Welcome to Lab Life with the Air Force Research Laboratory.
Hi, I'm Michelle.
And I'm Kenneth.
Hello, folks. Today we are joined by Dr. Rajesh Naik and Dr. Justin Gallivan to discuss AFRL's Biotech Grand Challenge, Microbes, and how to make the perfect cup of coffee.
In 3, 2, 1.
Dr. Naik, welcome back to the podcast. And Dr. Gallivan, welcome to the podcast for the first time.
Thank you.
Thank you.
We thought we'd kick off this episode talking about coffee for a little bit. Is everyone well caffeinated? What are you drinking today?
Unfortunately, I'm not drinking my coffee today. I had an espresso in the morning. Now I'm having just Indian tea. So chai tea, as we call it.
Sounds good.
I've got sparkling water. I've reached the age where I can drink coffee in the morning, but can't really stay caffeinated throughout the day, unfortunately.
But Justin, you're a connoisseur of coffee, aren't you?
Yeah, I drink the good stuff. I mean, life is too short to be wasting time on bad coffee.
What was the contraption you showed us one time on a meeting, how you can actually make some really great coffee? What was that?
Well, I have several contraptions at home. Probably the most elaborate is a siphon, which involves heating water in a vessel until it boils and the steam forces the water up into an upper chamber of that vessel where you add the coffee, and the coffee brews for a set period of time, and then the brewed coffee goes back down through the filter into the original vessel. It's a little hard to describe in words, but it's really cool to watch. And it's elaborate and fun to look at, but most days I just do a pour over with a Chemex.
It's a little fancier than my old percolator that I have at the house with some Highlander grog, though. That's my favorite. Got to have a little flavor with my coffee.
Well, definitely better than a Keurig.
I was just wondering then, like in terms of this device that you had, where did you hear about this, or where did you get that idea? Is there like a great like coffee connoisseur club we should know about?
So I actually used to be a professor of chemistry at Emory University in Atlanta, and not too far from my office was a wonderful coffee shop, so an independent coffee shop where the baristas there were really terrific. I got to be good friends with them and learn a lot about the science behind coffee, as well as just sort of the usual things about brewing it. But we really learned about where coffee originates from, what makes good quality coffee, what are some of the factors that are important if you're going to prepare a good cup. And you'd be amazed at how well you can coax certain flavors out of coffee. For instance, I used to feel it was necessary to drink coffee with milk, and it was just because it was so sensitive, so bitter. But when I learned how to make coffee correctly, I learned that I didn't actually need the milk. So I actually drink coffee black now. But every once in a while, I will go with a cappuccino, or a cortado, actually, is more my speed.
Well, I know whatever Ken drinks, he usually needs a lot of sugar throughout his day, so that might be his speed too. But you touched on something like origin of coffee, and in a different life, before you were a senior scientist in our human performance wing, you were a program manager at DARPA, and you had research related to tracking where coffee beans originated based on microbes on the surface, I assume, of the coffee beans. What's that about?
Well, that's correct. So the goal, of course, wasn't specifically coffee, although it provided a really good use case. So the problem that we're interested in addressing is determining the origin of particular items. And it turns out that both you and me and everybody that's listening to this is covered in microbes, mostly bacteria, many viruses, many of which are innocuous. So we're not thinking COVID today. But those microbes imprint upon us, and they differ in different environments. And it turns out that with the advent of high throughput DNA sequencing, we can essentially gather up the DNA from all of the microbes that are on a surface. We can run it through a DNA sequencer, and we can essentially identify what types of microbes they are. And because the microbial burden is different in different communities, there are differences. And it turns out that those can reflect geography. So for instance, if I were to take an object that I had found in San Francisco and I were to swab it, sequence the DNA, I would be able to start getting a picture of what objects in the San Francisco area look like. Whereas if I took an object that I picked up here in Dayton, it would have its own microbial signature. And it turns out that those signatures have not just a specific location, but also a regionality. So the microbes that thrive, say, in South Carolina look more like the ones that thrive in North Carolina than those that exist in Hawaii. So you can start building, using DNA sequencing, a map of the world. And then if you receive objects that have traveled through a certain part of the world, you can often identify not only where they came from, but also the places that they transited through to get to you. Now, why is this of interest in the coffee industry? Well, one of the things that is important in a lot of contexts is validating a supply chain. So if we want to know, did something come from where a vendor says it did? Is a part original? One way to do that would be to sample the microbes in the known factory where we think it should have come from, and then subsequently sample the products when it comes to us. And we can determine whether or not it came from a legitimate source. And obviously, this is a huge problem, not just within the Department of Defense, but also within business in general. For instance, if you buy something that you believe is being made in a particular factory, you don't want to find out that it is being made by slave labor in a different part of the world. And this type of technology can provide some insight into that. Now, the interesting thing about coffee as a test case is that purchasers will pay a premium for coffee that comes from a particular location, often even just a single farm. So if you want to validate whether something came from a single farm, and that farm is not only the one that says they're sending it, but it's that part of the world, it's the ground truth correct thing, as opposed to something that came from someplace completely different part of the world, or blends of beans, some of which might be of high quality, some of which are less quality. So you're not actually getting the real, sort of the real deal. And the microbial signatures are really good. So we work with some partners within industry to sort of determine whether or not this technology could be used to determine the provenance of objects. And it turns out it actually works pretty well. And it, again, all comes back to this idea that we and everything around us are covered in microbes.
From what it sounds like then, like you said that there's such an accurate telling of where objects come from. Is there like a map that you guys reference then? Something like the microbes of the world that we could have in classrooms to know where certain objects may come from?
So it turns out, yeah, the challenge there, of course, is sampling, right? So in order to get that map, you have to physically go to those places. Now, if you're making a block by block map of the city of San Francisco, that's much more straightforward than trying to map the entire universe. So in cases where you can't map the entire universe, one of the things that you can ask is, did two things come from the same place? So even if I don't know where they came from, I can say that they likely came from the same place. So if I have an authentic sample that came to me that I know to be authentic, and then I receive a second sample, I can determine whether or not those likely came from the same spot, even if I don't know what the original spot was.
And I'm just imagining the next time I turn on CSI that someone's just going to have a microbe map and like try to pin the killer that they were definitely in this location because, I don't know, something's on their shoe or their gloves or something like that. Can that happen?
Yeah. So there are actually, as you know, we do DNA testing, fiber testing all the time, recognizing that a hair sample is there. And there's absolutely no technical reason why the presence of certain microbes can't be used in a similar way. As with any of those tests, there's a confidence level, right? That we can say there's a 1 in a million chance that this person did it, or a 1 in 7 billion chance. And, I think the science is at a stage now where we're still working through that. And of course, there are attendant legal issues that are outside of my bailiwick. But I suspect that because the technology is becoming ubiquitous, the cost is going down, that it's going to be part of the armament for law enforcement.
And I think, Justin, you hit the nail on the head there. Is the cost of doing the sequencing and the analytics, becoming more mainstream, more manageable in terms of cost and expertise. A lot of the systems have become very automated. Now more people are getting into it, right? Just not the highly educated, molecular biologist or bioinformaticist. It's becoming more democratic, where you have more people getting into this. So it's a great thing, and that's why I think the Air Force is interested in this as well, because we could use some of those processes to do provenance of things that we are interested in, provenance of things that we're interested within the DoD. It could be electronic components, it could be other things, because as you were saying, going back to coffee example, right, the beans coming from all these different parts of the world, you can trace it back, so you can figure out the authenticity of the material. So you have supply chain integrity and security built in using some of these tools.
So it sounds like from what you're saying, Dr. Naik, that a major evolution in this is not only really us getting out there, but it's going to be the cost effectiveness of this. So in the future, once you said it's more accessible to people, like this could really change to a much larger enterprise for people.
Right, absolutely. And you're seeing a lot of DIYs and other things in sort of the synthetic biology area happening because of the cost being driven down, right? And people do this in your garages and basements now. And, you can buy a small device, from companies that look almost as small as your cell phone that can do sequencing for a couple $1,000. Yeah, maybe it's not high quality, but at least it's a start, right? And so,
to put that in perspective, when I was a graduate student an awful long time ago, if we wanted to sequence 300 base pairs of DNA, that would cost about $7. And now we're getting the possibility of whole genome sequences of humans for under $1,000, and the costs keep going down. So we're talking 300 bases for $7 versus 3 billion bases for about $1,000. So if you do the math, the cost reduction is incredible.
And something we'd like to do then, like now that we have a good idea of like what microbes could be capable of, at least in a sequencing scale, can you kind of describe to our audience who may not be as familiar just what they are?
So microbes are essentially anything that's small and of biological origin. So viruses are technically microbes, but today we're just going to focus primarily on bacteria. So bacteria are small in the sense that we can't see them with our eyes, but if you look under a relatively inexpensive microscope, you can see bacteria. So they're about 1,000th of a millimeter across. So small, but not super small. So, size is is relative, to an astrophysicist versus a biologist.
No, and that's a great point. And that's something like we wanted to ask as well, because I know a lot of people hear microbes, that can spark a lot of different ideas. And I assume the ones you're working with then are like anything that's not too dangerous, or does that kind of just depend on the nature of the work?
Yeah, so microbes get a bad rap because when we think about, sort of in the public vernacular, we think, oh, I got E. coli poisoning or salmonella or something like that. But the vast majority of microbes have evolved along with us for, thousands of years. So, and they coexist. So they on our skin, they're there. Even if you wipe with soap, they come back. Within our guts, the microbes help digest your food, so they can break down some of the more complex things that we eat into smaller components that can get absorbed by our body. It turns out that there are lots of interactions with the microbes that live in your gut and your immune system. And it turns out that your gut actually has lots and lots of synapses, connections to your brain. So there's something called the gut-brain axis. So when you think about your gut feelings, that actually is, more than just an expression. What goes on in your gut can actually have an impact on your mood, for example. So then if you take it a step further, that if you have a gut-brain axis and that the microbes are interacting with the gut, there are hypotheses out there that suggest that the microbes that live in you can actually affect things like your mood, for example.
So you're saying when I eat chocolate cake, that the microbes in the chocolate cake do actually just make me feel better because of the brain gut axis.
Well, it may not. Chocolate cake. There's lots of other things that are good in chocolate cake that you're probably detecting, but there are compounds in chocolate like theobromine, which is going back to the original story. That's actually a very similar structure to caffeine, and that's one of the things that you find in dark chocolate that has been implicated in antioxidant properties and some of the reasons that it's good for you. So,
well,
eat
if
more
you guys.
dark chocolate.
Okay, I'll take that as an order. But if you guys have any studies you need volunteers for studying the microbes and other forms of dark chocolate, let me know. But a lot of our talk today, we've kicked it off with talking about microbes and coffee, but we want to dive into biotechnology. What is that field of study?
Oh, well, that is incredibly vast. So maybe I can break things down a little bit. So maybe in some of the contexts that we're interested in, it turns out that the microbes are incredibly useful, and we've known that for thousands of years, even if we didn't know the scientific explanation. So yeast are microbes, and if you want to bake bread or brew beer, that's using the action of microbes to convert sugars into carbon dioxide, which gives you foaminess, and it produces alcohol as a byproduct. So both obviously in brewing beer, we prize the alcohol component, but if you bake bread, you prize the rise that you get on the bread. And there is a little bit of alcohol produced in the process, but since we don't use quite as much sugar, that amount is low and it gets baked away. I actually have right now proofing in my oven is sourdough bread. So I got into that during the pandemic, but there's another example where you have wild yeasts and bacteria that are fermenting products and imparting new flavors because of the compounds they produce. Now, on an industrial scale, we use microbes to produce all sorts of things. So antibiotics are paradoxically or sort of curiously produced by microbes. So oftentimes antibiotics are the product of that one bacterium uses to fight against another bacterium. So we ask the one that produces it to produce it for us. We scale that up in a big vat and we can isolate antibiotics. So in general, biology is an amazing technology, right? So you can take, something encoded within within DNA, which is built of A's, T's, G's, and C's. Those are the basis in DNA, which you can start thinking about as being equivalent to ones and zeros that we use to write computer code. So what we've been able to get very good at over the last several decades, but it's really coming into its own now, is that, as we talked about earlier, we can read DNA through sequencing, and we can also write it. So that is to say, we go to our keyboard and say, I would like to produce this particular sequence of DNA, and then often using a robotic system, you can actually produce that. So you're really starting to essentially hack the operating system of organisms. So that opens up a huge amount of possibilities.
Yeah, and I think going back to biology becoming a technology, you can see sort of the booming biotechnology across the globe, right? Industries are popping up. Open any magazine today, you will see some reference biotechnology, right? Whether it's artificial meats, how do you develop sustainable processes for leather, for example. Now they're making artificial leather using a biosynthetic based approach or a synthetic biology approach. So lots of fascinating things that's happening, I think, in the biotechnology space. But more importantly is how do you bring this back to a military utility, right? That's why we are here, right? All the cool stuff that's going on the civilian facing side, But you also have, I think, here an opportunity to really influence, I think, military, needs, right? There's some gaps. There are opportunities to come up with new ways to build materials. There are opportunities to enhance the physical and cognitive resilience of our warfighters. There could be ways to make precursor materials, biofuels for maybe the future hypersonic vehicle or something of that sort. I think can all be enabled by biotechnology. Because what biotechnology is an enabler, right? It's not a product by itself, but it's an enabler that allows you to develop, a product that can be then introduced into a commercial application or a military application. And that's why recently we just had 3 grand challenges that was awarded to the external community, looking to see our industrial partners from the US as well as internationally to solve some of our problems and mature to a higher TRL level. We within the lab have done some initial technology demonstrations with some of these approaches, but now we need to have the robust industrial support that we need to now mature that technology so it can make a product that then the Air Force can pull into its inventory. Right? So those three new biotechnology challenges that AFRL sponsored is exactly doing that, is maturing some of those observations that we've done in the lab, now really maturing that technology to create a product that would enable or provide some game-changing capabilities for the Department of Defense and for the Air Force.
Yeah. And for our listeners, when you say to a higher TRL level, you're not saying total request live with Carson Daly. You're saying technical readiness level.
That's right. I don't know what the other thing means, Michelle, but yes, it's technology readiness.
This is very generational.
Hopefully some of our listeners understand. I mean, I didn't even have cable as a child, but TRL means something else different to me. But going on there, so back to the actual, podcast, then when we're talking that there are different focus areas in our Biotechnology Grand Challenge, what are the focus areas this year?
So this year, in the Grand Challenge, there are 3 areas that we've identified. One is building materials for high temperature resins, so precursor materials for high temperature resins. There's some unique challenges there that synthetic biology can address, so that's one. The second area is in the area of biofuels, right? Because if you want high energy density fuels, there's some challenges with existing processes. And there is a way to develop some precursor molecules that exhibit some unique properties using synthetic biology approaches, right? That could be much more cost effective than using conventional methods. And the third one is on engineering the gut microbiome to enhance physical resiliency. So how can we improve physical and cognitive performance in warfighters by engineering microbiomes? So maybe sometime in the future, you can go buy a probiotic that you buy currently in the grocery store maybe, and you drink it before a mission. So it provides you with additional physical and cognitive protection in our operational environment. So those are some of the 3 areas that we're focusing on this time around.
So I know, harkening back to the last podcast we spoke with you with, Dr. Naik, would this other probiotic option maybe be fighting hangovers for your, in the gut biome?
I think you can buy that, right, Justin? I think
there is a company that claims that they can cure hangovers. I was at a meeting in San Francisco where there's a lot of folks that are into nootropics and these, trying to improve your own performance. And these guys swore by it. They were handing out samples saying it works. I generally avoid taking biological samples that some random dude hands to me. So,
that's a safe bet.
But yes, there is the possibility, and there it is. It certainly is within the realm of the possible. So, here's an observation that sometimes individuals have different responses to the pharmaceuticals they take. And it can in part be due to the fact that if the microbes in your gut are helping process those or degrading them. If you have a set that breaks down something more quickly than mine, I'll get a higher dose, effective dose of the drug because your body has broken it down more. And this is something that we didn't even appreciate years ago. I mean, these microbes have lived with us, we've co-evolved as humans to have them around. So people that eat a Western diet have different microbial profiles than those that eat different kinds of diets. But in general, we didn't appreciate the scale to which these microbes were there because we couldn't look. And DNA sequencing, as we talked about earlier, really provides that window into figuring out who's there. And now I think one of the big questions is, well, what are they doing? And that's part of current research.
Something I kind of remember from a conversation was that one of you said that obviously there's ethics around changing a human's DNA, but we can mess with the stuff that's on us is something that we can do with microbes.
That's correct. So it is part of Department of Defense policy and many others throughout the United States that we won't edit a human genome, and certainly not for non-therapeutic purposes. So we're not going to create superheroes. Even though we might like to, it is we're not there yet, and it's a bad idea. That said, these microbes that are living on us, we could imagine endowing them with some extra capabilities. So, for example, could I have the microbes that are on my skin if they were to come in contact with something that was an agent that I want to detect, something bad. And all of a sudden I start feeling a tingling, or I see a color change. Or better yet, what if those microbes could recognize chemical agents and break them down? So providing essentially a second skin or a form of biological armor. And all of this is within the realm of the possible. And then the advantage is it's fully reversible. So, you can just wash with soap or scrub everything off, and it doesn't actually have any permanent effects. But if you're imagining offering some protection over a period of time for an airman on a mission, it provides a particularly interesting potential solution.
And I'm just trying to get my head around that, because the capabilities are it's awesome. Like, to think about just what we could be capable of. I know we had talked about, kind of diving into what you said there, from early detection on your skin. It almost sounds like Frodo with Sting in Lord of the Rings. Like, it glows blue when there's danger, like orcs nearby. And that's so cool. Like, so other capabilities then, like you said, there could be armor, for instance, or is there a way to detect radiation, or is there really, what are the limits to this, at least as of what we understand right now?
And I think that's a potential, right? There's some studies that have shown that you could develop microbes that are living materials, right, that can detect different things in the environment. It could be in your skin, it could be on a wall of a building, it could be on one of our aircrafts or UAVs. So think about it going through a plume of things, and it gets activated, and it could be an artificial skin. In fact, there was a recent article just came out on kombucha, the tea that you buy in grocery store. It's a fermented product, and I think, correct me if I'm not wrong, Justin, it's actually a cellulose. I think that's made by the microbes in there. But they use that process as a way to embed within that film that's made, that map that's made out of cellulose, cells that can respond to a chemical pollutant. So it'll change color when there's some nasty chemical in the environment, right? So you could think of that as if you apply that bring that into our environments. Maybe now, where if it's a nasty chemical that is released, maybe it lights up. So you really don't have to put people in harm's way. You don't have to send your assets in. You just have a living material that will sense it, and it will provide a signal that you can detect through very standoff approaches. Now, there might be some sensors that you can get through electronic processes, but I think what biology gives you, it gives an additional dimension that maybe a electronic-based sensor cannot give you, and the ability for it to stand and survive extended periods of time be truly a living material. I think that is what's unique and I think exciting.
Yeah, this is the 21st century version of canaries in the coal mine.
Coal
right?
mine, yes.
Yeah, absolutely. And I think you talked about it beforehand, but you said a major way that warfighters, for instance, like kind of tying this back to the Air Force, let's say like the idea that we do have these microbes on the skin that could you detect or help you in the environment. Would this be topical then, like something you'd put on, or like you said, something you would take instead for the gut biome? Like, or I guess it really depends on the project.
Exactly. So you could imagine, something that you would just apply, sort of the equivalent of sunscreen, for example. And you can even imagine having microbes that are actively producing sunscreen, so you don't have to put it on. Now, that's great for a variety of reasons. As somebody who I have Irish skin, so I burn a lot. It would be nice not to have to think about it. And if you think about our warfighters, they have a lot of burden, right? They have to remember to put the sunscreen on. You have to remember to carry it. That adds excess weight, something that, maybe instead of carrying that sunscreen, you can carry an extra battery because you have more space and you've offloaded some of the burden. So not only just the burden of carrying something, but the cognitive burden of remembering, did I put this on? So if your skin is essentially, or the microbes on it are providing that capability that you don't have to think about it, and it doesn't add any weight, that's potentially very attractive.
That's just thinking of some of those applications outside of the airmen. What else could like these microbes or these like a lot of these technologies do for? Can they do things for like aircraft or other technological pieces, or is this more centered around the airmen themselves?
Right. I think you can think of what do microbes do in a gut or a skin or even a nasal cavities? They provide some layer of protection, right? They keep the bad actors away and sort of provides that perfect balance, right, between good health versus getting infected by bad things. So think about if there's a way to engineer some of those things that can prevent maybe corrosion in our aircraft systems or protect our fuels from getting degraded. I think there's so many things you could think about as applications, right? And one of the other part is also, the Air Force and some of the work that we do is in support of the aeromedical evac mission for the DoD, right? You can think of maybe there might be smart bandages where, you can come up with that might have some of this biologically synthesized materials, or could be an engineered microbe that can release compounds in response to given stimuli to enhance wound healing or do all sorts of neat things. I think the possibilities are endless. I think you're going to see more and more of this on microbes being engineered to do lots of things and things that we might not even have thought of. I think it's going to be possible in the future. That's been enabled by advances in just not DNA sequencing, but in understanding better pathways and how do things interact. I think the science is exploding in that space.
It's something where I kind of tie back to what we were talking about earlier with the future of what this looks like. Like you both mentioned, we're on the forefront of some really, really cool stuff and already diving into some very interesting topics. What can we look forward to seeing, especially tying back to the grand challenge we have here? Maybe technologies they'll be working on that maybe you know these challenges could be promoting, or at least what they can kind of kickstart here through this year and beyond.
I am really attracted by this idea that, DNA is just a way to express information chemically and that we can now start programming it. So one of the projects that I worked on at DARPA was asking the question, can we program an organism to undergo a particular plan of development? So nature has shown this, right? I can take a tiny seed, I can plant it in the ground, and I can get a tree. And although there's variation between the trees, a particular species has the same size and shape, just like people, right? We vary within some sort of limits, but all of us were programmed by a little bit of DNA from mom and a little bit of DNA from dad. Now, you think about that tree, right? A lot of what we use trees for is we want to get dimensional lumber. and it takes a long time to grow a tree. And then we take this beautiful living thing and that can take CO2 out of the air and release oxygen, and we cut it down and we kiln dry it, and we produce a 2x4 that is useful, but immediately is inert. It doesn't do anything other than take up space and bear loads, things like that. It's already on the path to degradation. So what if I could program a plant cell to undergo development such that it grows to a particular size and shape? So instead of growing trees, I go 2x4s. And what if those 2x4s maintain some of the properties of the plant? So the ability to heal itself, right? Just like a human, if we get cut, we heal, we get a scar. It would be great if you had materials that could do the same thing, that could repair themselves. And if you start thinking about that, the amazing things that biology does, and start asking, how can we harness that? And all of that is within the realm of possibility. It's incredibly difficult. I mean, I'm not going to lie to you. This is not coming to store shelves this Christmas season. Certainly you can write the problem down and say, these are things that what seemed like science fiction and have gone from the perhaps impossible to the merely improbable. And that's what we scientists do, is try to explore the art of the possible and to bring them into the realm of the practical so that we can deliver solutions to the Air Force and the DoD.
And I think that's key, right? Is the possibilities are endless, but how do you work on things that are most beneficial to the Department of Defense, right? And some of the challenges, certainly in human performance, in materials, and other aspects. I think there's a lot of potential for biotechnology. And as an enterprise within AFRL, we are working. We have a portfolio, what we call the biotechnology portfolio across AFRL, where we're working across different TDs, including AFOSR, including industrial partners, international partners, other services, on working on problems that are Air Force specific, and have opportunities for us to leverage, some of these advancements that you see, because some of the things that Justin has been talking about, a lot of those things are happening on the outside, not necessarily within the Department of Defense fences. So we should be able to, reach out, leverage some of those advances and apply it towards more DoD Air Force centric problems. And that's what we're doing, right? We are training our workforce. We're having smart buyers of practitioners of biology. But we have to figure out also at the same time, how do I take some of this cool stuff and apply it to some of the most challenging problems within the DoD? And we as a community have come together around this space, because this is one of the top priorities for the Department of Defense. It's one of the modernization priority area for the DoD. Under the OSD modernization priority areas, biotech ranks as one of them. So again, it's an opportunity to develop some unique Air Force solutions. I think we have opportunities to do that and show how biotechnology can provide a game-changing or paradigm-shifting capability for the Air Force. And the onus is on us to do that. And I think we have a very strong workforce, a very smart workforce in the organization that is capable of delivering on that ask. I think there's also several press release articles that's open. If you go to our main website, we just recently released one on the 3 challenges that provide some input. They can reach out. Any of our listeners who are interested, reach out, and we'll be happy to have a conversation.
Maybe get some more chai tea and coffee, because I have so many crazy applications, and maybe it's just, some of the science fiction Marvel movies I've watched and series that I'm just like, but hey, would this work, to make someone a superhuman or something like that. But anyway, I will close out our podcast trying to see how nerdy Dr. Gallivan is. We know he's super nerd about coffee, but you said you were making your own sourdough bread, right? Have you gone so far as start to grind your own yeast so that you can with different types of yeast from different fruits or whatever in your bread?
I have not. my brother and I are in sort of this competition where we keep going back in the process. And at some point I wonder, am I going to have a patch of wheat growing in the backyard so I can go fully artisan? I don't think so. To tie it back to some of the microbes that are found naturally in the environment, there is, and I can tell this story because it's almost 5 o'clock and I'm going to go drink a Guinness. But there's a story that that may be apocryphal, I don't know, where somebody had written to the head brewmaster at a brewery and asked for a sample of the yeast that they used to brew their beer, which was really good. And then they replied, and this is long ago, where you actually got a letter back instead of an email. And the story goes that the person was able to swab the letter, isolate the yeasts that were floating around in the air of that brewery, and be able to brew that particular type of beverage. So I don't know if it's true, but it is a good story, and I think it's certainly within the realm of the possibility.
Judged by what we talked about today, that sounds like it's in the realm of possible, at least. So that's really cool. We want to thank you both so much for taking part. It's been such a pleasure, and I'm sure we'll both speak to you again soon enough.
Thank you. Thank you, Michelle.
Great. Thank you both. Looking forward to it. Take care.
Make sure to follow us on social media at Facebook, Twitter, LinkedIn, Instagram, and YouTube at AF Research Lab. And remember, stay curious.
Logging off.